Good morning friends. Today we will discuss about the stages of early embryonic development. Fertilization is defined as a process where gametes, an egg and a sperm join together to form a full zygote. Both the sperm and egg contain a single set of 23 chromosomes coming together to form 46 in the final zygote. To make sure that the resulting embryo only ends up with a single set of 46 chromosomes, only one sperm must join together with a single egg. In the mammals, the egg cell is preserved by an extracellular matrix made mainly of a group of glycoproteins called zona pellucida. Once a sperm cell is able to bind with the zona pellucida layer, a cascade of biochemical events takes place, the acrosomal reaction. In the placental mammals, the acrosome consists of digestive enzymes that are used to initiate the breakdown of the extracellular matrix surrounding the egg, that allowing the cell membrane of the sperm to fuse with the egg. The joining of these two cellular membranes forms an opening in which the sperm cell nucleus can be transferred into the center of the ovum, where the nucleus membrane of both the sperm and egg cells begins to degrade, with the two haploid genomes coming together to form a singular diploid genome. The next is the cleavage and blastula stage. The initial growth stages of multicellular organisms starts with the zygote cell, which then undergoes fast cell division to form the initial cell cluster or blastula. This rapid division of cells is also known as the process of cleavage. Once the cleavage process has produced well over 100 cells, the developing embryos is called a blastula. This blastula is typically a spherical layer of cells, otherwise known as the blastoderm, which surrounds a fluid-filled cavity, the blastocyst. Mammalian embryos form a new structure termed the blastocyst at this stage, which can be characterized by a mass within the cells that is obviously distinct from the other outer blastula region. During the rapid cleavage process, the cells have been found to divide, with increasing the total mass, meaning that this process is simply one large single-celled zygote that divides itself into several smaller cells. Each of these cells within the blastula can be called as blastomere. The cleavage specialization stage. The type of cleavage that occurs within the developing embryo is dependent upon the volume of yolk that forms inside the egg itself. In placental mammals, which include human beings, where nourishment is solely provided by the mother's own body and blood supply, the eggs only have a very small volume of yolk and thus undergo a type of cleavage called a holoblastic cleavage. Other species of animals undergo a process called meroblastic cleavage. For example, birds that have more yolk inside the egg sac that is used to nourish the developing embryo. In mammalian embryos, the blastula develops further to form a blastocyst in the next stage of its initial development. At this stage, the cells inside the blastula begin to arrange themselves into two separate layers, an outer layer termed the trophoblast and the inner layer known as cell mass. The inner cell mass can also be known as the embryoblast, with this mass of cells continuing to develop and form the full embryo. At this stage of the development process, the inner cell mass has been found to consist of the embryonic stem cells that will soon come to differentiate into the many different cell types that are needed by the organisms to function. The next is the gastrulation stage. In this, the development of embryos is the basic conception of the body plan. The cells within the blastula structure rearrange themselves to finally form three distinct layers of cells in a process known as gastrulation. During this stage, the blastula proceeds to fold in upon itself to form those three distinct layers of embryonic cells. Each of these new layers is termed as a germ layer, with each germ layer differentiating into a different organ system within the body. The three germ layers are called as the mesoderm, endoderm, and the ectoderm. The ectoderm initiates the formation of the nervous system and the layers of skin. The mesoderm germ layer activates the formation of muscle cells and connective tissues inside the body. Finally, the endoderm. The endoderm layer starts the formation of columnar cells that can be found inside the digestive system as well as many other internal organs. Once these three layers have become fully established and start to develop separately, the body begins to form and the embryo becomes a fetus. There are many structural genes utilized in this specialization process where if there are any mutations, parts of the body can grow in the wrong place. For example, organs growing outside of the body instead of under the skin, or it can cause miscarriage of the embryo. Thank you. Please like, comment and subscribe. And if you have any question, then you can ask your question in the comment box.